Okay, today we are going to finish our lecture on sleep. We have more slides than I realized we have, but I hope we'll finish that and have time for you at least to start talking about your homework. I think most of the homework discussion for the sleep chapter is going to have to, unfortunately, go to Wednesday, but we'll see. See what we do today. And then the plan is probably we're going to start that mental disorder chapter on Wednesday, is currently what I'm thinking. That is probably because it is completely from scratch and just the way my life is going, it's going to be in parts. So there will be like multiple versions or multiple copies of the slides as we sort of work through, maybe 10 at a time, maybe 12 at a time, as we work through that chapter. But that is, you can expect those to hopefully start appearing Tuesday night or so. I'm going to try to get going on that tonight. Also, as a reminder, your term papers are due Wednesday at midnight, and that's the only thing that's due Wednesday. Trying to think. I don't anticipate anything due the following Wednesday either. So, because basically we're going to meet on Wednesday, we're going to do our song and dance here. Then you basically have the week off because of Easter break, because we're going to be missing Friday and then Monday, and then we're back on Wednesday. When we're back on Wednesday, we'll be kicking into talking about the mental disorder chapter full force. We could potentially have a quiz like maybe Friday on the sleep chapter, but that would be, you know, sometime yet. I'll try to nail that down in my head. That would be the last quiz if we do that last quiz. Unless you guys really don't want it, but really? Some people really like quizzes. It kind of gives you a chance to catch yourself and refresh up after the break and all that stuff. I'll think about it. We'll see where we're going. We have had a lot of quizzes, but that's a good thing usually. So any questions about anything? Okay. All right. Well, then, this is more or less where we left off. So we were talking about, starting to talk about some of the neurological mechanisms that go on that cause us to sleep. So like, how do we sleep is kind of the question now. So that's what we're going to be trying to understand today for this first little bit. And one of the ways that we can try to understand the mechanisms of sleep and what brain areas are involved in different types of sleep is by using MRIs. MRIs are an imaging technology that basically are responding to where water is going. And so as you start to increase the usage of a brain area, you vasodilate blood vessels leading into that area, which causes more blood flow to go into that area to help basically to nourish these brain cells that are increasing their activity rate. And as a result, by tracking that, you can get a pretty good idea of what parts of the brain seem to be particularly active at different times or in different conditions. So these figures that are up here are trying to show you, I changed the labels here around a little bit just to make it a little bit more clear, I hope. What they're doing is they're comparing, these are comparison figures, comparing activity when you are in REM sleep versus non-REM sleep. And so the way it would work is that those areas that are sort of in red and yellow in here are showing increased activity during REM sleep, whereas areas that are especially in sort of the deep blue, those areas are showing decreased activity during REM sleep. So these are explicitly comparisons. These are sort of comparing at two different states. They're not just like a snapshot image of what's happening when you're dreaming, for example. Similarly over here, this is a comparison of REM sleep versus awake. So again, these areas over here would be shown as having more activity, and these areas would be shown as having less activity during sleep, during REM sleep, versus when you're awake. So given all that, what would you say, I mean, if you start making, I got some things written up here already, but if we just start comparing these, like what do you see as being different? Like what brain areas, can you see which brain areas are lighting up as being a bit more active? Just to kind of give you a reference, I think the way MRIs are looking, you're usually looking from the bottom up. That's kind of how they, just by convention, that's how they orient them. So we're looking for sort of from the bottom up, and we got a slice of the brain kind of like around here, like so not at the top, but you know, right around this area. So we're getting a lot of cortical area out here. This would be like deep telencephalon. This would be like thalamus in there. That might even be like, I don't know, third ventricle. I'm not sure. So this is anterior, this is posterior, lateral. So like what areas are showing up to you as Like, can you tell what areas are lighting up as being particularly active during REM sleep versus non-REM sleep? What do you see? Yeah, what do you see? Yeah, yeah. Right. You can kind of see it's lighting up here. I mean, I'm pretty sure that's thalamus. It's in the right spot anyway, and it's about the right shape to be thalamus. And it seems like it's lighting up as being pretty active. You're seeing a lot of these sort of basal central cerebrum areas seem to be lighting up as being pretty active. The thought is actually a lot of this is limbic system that's sort of in this area that's lighting up, which we're probably not going to do that much of the emotional chapter. I might reference it a few times in the mental disorder chapter, but the limbic system is sort of this loop or series of brain structures that seem to be involved in our emotional centers. So emotional centers seem to be upregulated during REM sleep. I think it's kind of interesting, some of the areas that are shut down, like that's occipital lobe back there. So that's basically our vision center back here. And it's pretty much shut down during REM sleep, which is kind of interesting because a lot of people have really vivid sort of sensory visual images in their minds that they're experiencing during sleep. And they'll report on these things like crazy things that they saw. But that visual, like the primary visual cortex at least, the area receiving information from your eyes, doesn't seem to be particularly active. What is active, on the other hand, would be areas kind of up here, and then it would be actually sort of deeper into the screen, getting up into the parietal lobe cortex. Those areas seem to be like pretty active. So you're getting some of the association areas associated with sleep, so some of these multimodal association areas back in the parietal lobe, but not the immediate sensory processing areas. So we have sort of conscious perception of vision from these association cortices, and we're like thinking we're seeing things, but it's not actually coming from the eyes and being processed in our normal visual centers. And there's similar sorts of studies or similar sorts of situations in other areas to that, where we experience loss of sensory information, but it seems like it must be coming from these multimodal association areas that are kind of doing advanced processing and integration, as opposed to like the sort of root basic processing centers, which kind of makes sense. I mean, you're not actually seeing these things. You're sort of, it's all imagining and in your head.
So would the same area light up when you're having like hallucinations?
So that's a really good question. I don't know. That's a great question. I think you could certainly predict that. You could probably predict the opposite too, but I think you could expect sometimes hallucinations would come from that area. It would make sense. And it would be the same areas that are being excited in a dream state. These areas here are probably getting into those sort of association cortices areas. It's a great question. Okay, so this is kind of a, well, I did say these things. So this is, the next slide is sort of summarizing some of these things that we're seeing. So going from non-REM to REM sleep, so the REM areas would be red and yellow. We see a fairly substantial explosion of cortical activity, especially in some of the sensory association areas, not the primary centers that are doing like immediate processing from the retina, but the sort of next level of processing areas, sort of getting away from that back of the occipital lobe and getting up into the parietal cortex. Those areas seem to be running pretty aggressively. So we're kind of creating our sensory, the sensory experiences we're getting are probably coming from those sensory association areas as opposed to primary visual cortex or primary auditory or olfactory cortices out in the temporal lobe. And again, lots and lots of activity is present within our emotional centers as well, which is also kind of an interesting thing. It does highlight that dreams are often filled with all kinds of emotions and anxieties and whatnot. I know I have my recurrent nightmare that it is gradually transitioned over the course of my time. When I was a student, the nightmare was that I forgot to show up to take some exam or forgot to take a course all semester. And now the nightmare is that I forgot to teach a course all semester. And it's like the exam week, I'm like, oh shit, I'm going to get fired. So these things happen to all of us. These seem to be, like emotional centers seem to be heavily upregulated during REM sleep. Over here, what we're doing is comparing REM sleep versus non-REM, versus being awake. And I think that's also interesting. So, when you look at it, EEG, of a person that's experiencing REM sleep, it's almost imperceptibly different from the EEG of someone that's awake, despite that clearly there's massive differences in the behavior of the individuals. And what this is showing is that we have substantial upregulation during REM sleep, even above awake levels, of some of these, again, sort of these multimodal association areas back here. But this area, like, what's all that? Like, what lobe is that? You guys know. What is it? Frontal lobe. Yeah, so this is all frontal lobe cortex area and you know, some of the deeper areas within frontal lobe. These areas seem to be heavily downregulated. And at least, especially when you get into the anterior frontal lobe, this is where we're doing a lot of our like substantial sort of logical processing of thought and you know, active interpretation of what we're seeing. And those areas seem to be fairly substantially downregulated during sleep, especially during REM sleep versus when we're awake. So one of the arguments here is that we're getting a ton of sensory information, or at least we're sort of perceiving a ton of sensory information in our association cortices. And we're getting, at the same time, a downregulation in our ability to process that information. So like we're getting emotional and we're perceiving lots of things, but we don't have our frontal lobe to really make much sense of it, and so we have absurdity, is kind of the explanation. So those are some of the brain centers that are involved. How do we transition between these different areas? It seems as though a lot of the transition that causes us to move from, say, deep sleep into REM sleep, or REM sleep back into deep sleep, or sleep versus going back awake again, have to do with some of these large-scale modulatory, diffuse modulatory neurotransmitter systems we were talking about last time. So these are norepinephrine coming from the locus coeruleus, the serotonin, which is coming from the Rafe nuclei, and then acetylcholine, which is coming from many centers, but some of these midbrain centers seem to be important in terms of originating these acetylcholine systems. So if you're going to transition into REM sleep from deep sleep, this is what this first bit is. So transitioning into REM sleep is probably a better, instead of during REM sleep. The idea is that you'll have most of your neurotransmitter systems will be basically, you know, kind of inactive as you're in deep sleep, your non-REM sleep. So we're kind of here. But to shift into REM sleep, what will happen is you'll have upregulation of the REM-on cells, they'll say. And these REM-on cells are basically acetylcholine-releasing neurons, so cholinergic neurons. So we have cholinergic neurons from the pons start to fire. These send signals to different parts of the brain, and they get your cortical areas excited. And so you get that those differences that we were seeing on the prior slide, where the limbic area and some areas of the cortex really start to show substantial increases in activity as you enter into REM states. There's another cool thing that happens at the same time in response to these cholinergic neurons. So as cholinergic neurons start firing, you also see spinal neurons begin to become inhibited, and you literally become paralyzed, where you're unable to move during these same periods. And this kind of is a cool thing, and this came up on the homework, so I don't want to dwell on it too long now. But the idea is that while you're experiencing all of these sort of sensory experiences, and you're not processing them very well, and you're not actually getting real sensory experiences, simultaneous to that, you will paralyze yourself, basically, and that will keep you from moving and acting out your impulses that exist within your dream, hopefully. That's the idea. So if that doesn't happen, sometimes you'll have what's called REM sleep behavior disorder, where people start to act out their dreams, which is technically not the same thing as like normal sleepwalking, which often happens during non-REM sleep, where people just kind of are pacing about because they don't have that direct inhibition normally, or this direct paralysis. But during REM sleep, we are literally paralyzed. To shift out of REM sleep, what seems to happen is you have a decrease in the firing of the REM-on cells, the cholinergic cells, but you also have an increase in firing in what are called REM-off cells, which are norepinephrine and serotonin. And if these really start firing like crazy, they'll cause you to wake up. But instead, just to switch from REM sleep to non-REM sleep, what seems to happen is they show a modest level of increased activity, and then they kind of taper off. But that increase in activity causes other brain areas to get excited other than the REM areas, and that seems to trigger you to shift back into a deeper sleep. The exact mechanism to me is a little bit funny, because these are excitatory, and these same neurons, when they all start firing in concert, will cause you to wake up. But in this case, they're causing you to shift out of REM sleep and get into a non-REM sleep mode again. In the literature, even in the papers I think I had you read, you'll see discussion of REM on and REM off cells. But this is really what they mean. Is they're talking mostly about cholinergic cells for REM on, and REM off would be norepinephrine and serotonin. And the reason, again, is that these cholinergic ones in particular, these types of cells tend to be really active in releasing lots of acetylcholine during REM events. Yeah? I think I have heard that at least that is a hypothesis for some of these interrupted sleep things. A lot of what I see on interrupted sleep is more to do, not so much, I haven't seen a lot on neurotransmitter level. I wouldn't be surprised that work is better developed than I know about. A lot of like interrupted sleep here I hear about is more like breathing problems and other kinds of things like that that disrupt sleep. But yeah, that would make sense to me, because if your norepinephrine and serotonin is what causes you to shift out of REM sleep, those are the same excitatory neurotransmitters that get going aggressively during wake periods. So that, I certainly would think that would be true, but I don't know the literature as well as I could. Good idea. Okay, so this seems to be this is kind of like at least a basic overview of some of the neurotransmitter systems that are behaving differently during sleep versus not sleep, and in particular during REM versus non-REM sleep. I think one thing that I do want to just briefly touch on is this notion of dreams and dreaming, because that is usually, we think, coincident with REM sleep, although the data are a little bit fuzzy in places about this. But I think it is really interesting because dreams, especially for some individuals, I mean, it's actually, I just talked about my recurrent nightmares, but for the most part, like, it's pretty rare that I remember dreams. I just don't. Whereas other people wake up and they have a story to tell, and they are hell-bent on telling the story to people too, because it's so interesting and they want to tell everyone. I just don't really remember my dreams, but I think I do have them. Like, I have this sort of vague notion that I had some dreams last night. I just don't remember any of them. So what are these dreams? Like, what is the point of all of this? And there's all kinds of writings and such that goes back centuries on the meaning and purpose of dreams. So, Freud was one of the first people that really pushed this, and he, really pushed dreams as this this mechanism by which our brains allow ourselves to achieve wishes or fantasy fulfillment outside of sort of the day-to-day operation of the waking world. And so there is sort of a whole literature on that kind of level of some sort of explicit mechanism for dreams. There's other explanations that that really emphasize some of these almost like restorative or memory processing types of functions that might be related to dreams. So while we may not realize it, our dreams may be allowing us to learn things better, and there is some evidence for this. So, for example, while you cannot listen to things probably while you're sleeping and pick up and learn information that way, and there's some discussion in your book about this idea of like pushing play on some recording and that talks to you about your anatomy course, and then you'd wake up the next day with knowledge about anatomy. It doesn't seem to work because we seem to cut off all of our sensory information during sleep periods. However, if you practice something prior to sleep, so if you're studying prior to going to sleep, or if you're practicing some sort of skill prior to going to sleep, there is data and evidence that indicates that you will somehow reinforce those memory pathways over the course of the sleep period, so that when you wake up, you will have better learned those things versus someone that has more time before going to sleep, but when they were studying, or somebody that doesn't get a good night's sleep. They don't seem to learn those things as well. So some of these studies, I say Tetris studies, one of my favorite studies that showed this was they had people, you guys remember Tetris? This is less of a popular thing. When I was a kid, this was like the rage, because it was like the game I had on my Game Boy, my original Game Boy. So you've got these little blocks, and you have to practice manipulating them spatially and then sticking them down to build this little structure. And if you get full lines, you clear out your space. If you have people practice playing a game like Tetris right before they go to sleep, and then you test them again the next day versus someone who didn't get a good night's sleep, or someone that practice Tetris, but several hours before going to sleep. And you control for things like how much time has passed. People that practice Tetris right before going to sleep actually performed a little bit better on Tetris the next day. And that was some indication that there's something about sleep, at least, that enhanced the ability to do the kind of spatial manipulations you have to do to succeed well in that game. There's anecdotal evidence from that same study that people, if you had them doing this for many days in a row, would report actually seeing blocks and things like in their dreams, and you know, there was actually sort of dream manifestations of some of the Tetris activities they were doing, and that might be indicative that it was the dreaming per se that might be helping with the memory and skill enhancement in this case. However, depriving REM sleep alone didn't seem to reduce improvement after sleep, so in some of these same studies. So it's not immediately clear that it's REM sleep per se that's really important, as opposed to just good, you know, good deep sleep that you're getting into as well. There is another explanation. Your book cites Hobson and McCarley. They call it the activation synthesis hypothesis. This is kind of the byproduct hypothesis, which for a lot of people is incredibly unsatisfying, but it is an explanation. The argument is that due to some of the activities that occur during REM sleep, that causes associations, sensory associations, and memories to be activated almost at random. And then our cortex tries to synthesize these into some sort of coherent narrative, just almost out of reflex, because that's what the cortex does, is it tries to process and synthesize and make sense of information. And that the idea here is that because this is more or less a random process, the dreams don't really have some specific function, which again, to a lot of people is a very unsatisfying idea. But, you know, it could be right. It could just be that this is just a happenstance for the way our brains are constructed, and it's not immediately clear that dreams have a specific function. But who knows, right? The jury is definitely still out. Some people get very upset about that. All right. What is left in the chapter is I want to talk a little bit about circadian rhythms. I know you guys went into some detail about clock genes, so we don't need to spend too much time on that because that showed up on the homework. But I do want to talk about circadian rhythms to kind of close out the chapter in the lecture. So circadian rhythms are day-to-day rhythms. So this is the cycling that we're doing on a day-to-day basis, and it includes a substantial period of sleep during that period, but it also includes lots of other activities. So this is a graph that's basically showing over a 2-day span. So starting right after waking, going through an awake period, a sleep period, awake period, and sleep period, what's happening by different metrics that you can record on a person. These include metrics about alertness, which certainly in this case sort of peaks, it looks like around 2 or 3 in the afternoon for this particular person they were processing. Each of yours might peak at a different time of day, and then crashes certainly during sleep because we are essentially unconscious and unaware of anything during sleep periods. But you can also track temperature, which also shows some substantial fluctuation and especially a substantial depression during nighttime periods, which I think is kind of cool. I mean, it's almost a degree down when we're sleeping versus during the day. Here's growth hormone release. So you get this huge surge of growth hormone that is released right at the onset of sleep. Here's cortisol. Cortisol is a stress hormone. So cortisol levels, generally, they're showing them actually declining over the course of the day and then increasing at night, which I think is pretty interesting because that doesn't seem consistent with my, my experiences. And here's just potassium levels, and they are seeming to fluctuate as well. So there's lots of things going on that indicate that we are cycling in substantial ways over the course of the day. It's not just about sort of going through the day, getting tired, going to sleep, and then waking up again. Our whole bodies, many of our physiological processes are not absolutely stable over the course of the day, and they cycle every day. Now, these cycles are usually pretty much held in lockstep with external stimuli. So, you know, each day the sun comes up and therefore it gets brighter. It tends to warm up over the course of the day. So there's an external cue as well as temperature cue. I think it's less important, but sort of the day-night cycle in particular seems to coincide very tightly with our sleep-wake behavior as well as these fluctuations that we're seeing in our circadian rhythms. I think it's an interesting question, though. You could ask, well, what's going on? What would happen if we didn't have these external stimuli? Would we continue to cycle in the same kind of manner? And so there's actually been a whole host of studies where people have tried to do just this, not only to animals, as I think I showed you earlier in this chapter and some of the figures I threw in, but also to doing this sort of thing in humans. So what they'll tend to do, I don't know how you pronounce this word. Anybody good at German? Zeitgebers? Is that right? Something like this. Somebody's got to be a German person. Like, took a lot of German in high school or something, which would be a lot more than I took. All right, we're going to say Zeitgebers. Zeitgebers are just time givers. So these are the cues that cause people to entrain their natural biological rhythms. And so there have been a host of studies that have tried to create environments where they deprive individuals of those kinds of external cues and then see what happens. So it turns out they have to go to real extreme lengths, because even subtle little things like the airflow in the area, you certainly have to deprive all external light sources, but airflow. When you provide meals to the person, like you can't just always provide meals at 8 o'clock in the morning to the person, because that's an external cue they can use to entrain their biological rhythms. So you have to construct these environments, usually deep underground, completely with all kinds of external stimuli removed, where the person might have control over a light switch, but they wouldn't have any sort of external cue as far as when they should turn that on or off. Food-wise, you'd probably have to provision them with food all the time, so they're manufacturing it themselves so that there's nobody influencing them. Certainly, you'd have to remove any clocks or anything else from there. And you basically watch what happens over the course of a month plus and see what happens to that person in terms of how their behavior shifts. So what you're seeing here, this is kind of a 2-day cycle. So it's a little bit confusing. I actually drew this dotted line down here over the figure from your book, because what you're seeing here is going from midnight to noon to back to midnight. So this is one day, and then this would be a second day. And again, they're just observing this over the course of every day. But they include the 2 days because you can see the person kind of drifts a bit. So initially, they're giving them normal day-night cycle cues. They're providing food at particular times a day, and so on. And the person is more or less maintaining a daily rhythm that's fairly consistent. I mean, it fluctuates a little bit from day to day, and I think everybody fluctuates a little bit from day to day. Basically, the underlying period, this is when they're sleeping. The dotted period is when the person's awake. The triangle is when they are at their lowest temperature of the day, is what the triangle is. And you can see those things don't necessarily always coincide with each other. So initially, they're getting lots of external cues. They're turning the lights on and off at certain times a day within this cave. But then they go into what they call a free running period, where they isolate them from all external cues, all external Zeitgebers. No time givers are provided. And you see what happens to the person is gradually the person seems to consistently shift, and this seems to be fairly true for most people. They tend to shift a little bit later and a little bit later and a little bit later every day. It's almost as if our sort of natural rhythm is to have about a 25-hour clock as opposed to a 24-hour clock. So you stay up a little bit later every single night, and you get up a little bit later every single morning in the absence of any kind of external cue. I also think it's pretty interesting that while temperature usually hits its low not too long before you wake up, in this case what's happening is this person is actually, their temperature low starts to get like right about the time they start dreaming, as they're doing sort of this free running, clock shifting sort of behavior during this no external cue period. Finally then, what they do over here is they start up the day again. So they start including a 24-hour day-night cycle. And at that point then, the person once again entrains on this sort of standard rhythm. They happen to do it in this experiment when the person basically had shifted a full 24 hours over the course of about 25 days. They could do it at other times though, like if they started giving the person at this point, this person is waking up now, and on day 21 here, this person's waking up pretty much at noon. If they started the clock on normally, it might take a few days, but the person would gradually reset back and use that time, that dawn versus dusk cycle to entrain their biological rhythms. And that tends to happen just sort of, without stress to do so. Like they're not requiring people when the lights come on, hey, it's time to get up. They're basically providing an environment, and the person will basically naturally see their brains shift their day-night cycles to match the external cues. But one of the big points here is that it's not just like once you turn off external cues, they become arrhythmic. they still maintain a rhythm. It's just not perfectly in cycle with the external environment anymore because there is no external cue for them to train on. So they just kind of shift back a little bit each day. That return to having a strict day-night cycle is called entrainment. So that's a specific term that you should know. That's just, entraining your biological rhythm to match up to the external cues. So if we have the ability to consistently cycle, even in the absence of cues, which is what's happening here, they're more or less cycling on a 24-hour rhythm. It's just not quite perfect. That means there must be internal mechanisms that allow us to do so. We must have an internal biological clock. And so there's been much investigation of how that actually works. And it seems as though it all revolves much around this particular nucleus of the hypothalamus called the suprachiasmatic nucleus. It's this guy right here. So if this whole thing is the hypothalamus, so this is the third ventricle in here, the walls of the third ventricle are the hypothalamus. Here's the optic chiasm. This is where this is associated with our optic nerves. Here's the pituitary gland. So all this area along here is hypothalamus. This one little spot in the hypothalamus is the suprachiasmatic nucleus. And this seems to be where our sort of most fundamental biological clock seems to be housed. And it's not just us. It's most mammals seem to have the same basic brain area that's in charge of processing and time and maintaining our biological rhythms. It does get direct input from our retina, so that's also consistent. We've got axons that lead directly to the suprachiasmatic nucleus without going back to the occipital lobe first. And even more interesting, this actually just appeared in this version of the textbook. So this is the 4th edition. The 3rd edition didn't have any information about this. So these are discoveries more or less that have happened over the last 5 years or so. It seems as though there's special sets of photoreceptive cells that are responsible for sending information to the suprachiasmatic nucleus. So most vision that we're sort of consciously aware of that goes to our occipital lobe you have light photons that are being picked up by rods or cones. Cones are daytime processing, color processing photoreceptors. Rods are twilight processing types of photoreceptors. Rods are completely saturated under this level of light, but under really dim conditions, they're the only thing that can run because cones aren't sensitive enough. Neither of those send information directly to the suprachiasmatic nucleus. Instead, there's a special set of cells, they're ganglion cells, so that we didn't even really recognize them as being photoreceptors initially. They're not in the same layer as all the other photoreceptors. These are ganglion cells that have a unique pigment. It's not rhodopsin, which is what's up in the rods and cones. It's melanopsin. But it seems to be responsible for absorbing light when there's light hitting, any kind of light, hitting your retina. And then it sends information from the retina then to the suprachiasmatic nucleus to say, hey, there's light right now, or hey, there's no light right now. And that's really all it's doing. It's not providing any other information about image or any other specific processing. Yeah. Yeah, so depends on what kind of blind and why the person is blind. I don't know for something like a retinal detachment if that would, it wouldn't surprise me if that would still be something they could pick up. Certainly the case where people who have brain damage to their occipital lobe that causes them to be functionally blind even though their eyes are still working, they wouldn't lose this ability, and they still have other kinds of interesting visual reflexes. Like they can track objects even though they can't see them, which is cool because that's superior colliculus is in charge of like tracking an object through space. And they may have occipital damage so they can't recognize or they don't visualize anything. They can't perceive any vision, but they could still like, track an object and walk around disruptions in the classroom without really like knowing it, which is kind of neat. But yeah, I mean, I think it does depend on why the person is blind. Certainly if the person has damage to their optic nerve, they wouldn't be. But yeah, I think that can be true. So it's kind of neat. There's this whole other set of cells that we didn't even really realize how to function like this. Most ganglion cells are about integrating information that's coming from photoreceptors and then passing that on up the optic nerve. They actually form the axons that send signals up the optic nerve. But here, you got a special set of ganglion cells that aren't really doing that. They have their own pigments, and they're processing and receiving light and sending that information to our clock gene, or to our suprachiasmatic nucleus to inform its clock centers. Here's some additional evidence that the suprachiasmatic nucleus is involved, beyond just the fact that it's getting information, this separate, weird, different kind of input from the retina. There's been ablation studies, which aren't fun studies, but these are ablation studies done in monkeys that show what happens when you ablate or destroy the suprachiasmatic nucleus. So you surgically go in, you destroy the suprachiasmatic nucleus, and you compare monkeys with a damaged suprachiasmatic nucleus, which are down here, to normal ones over the course of time. And here's what you end up seeing. So this is normal monkey, no damage to the suprachiasmatic nucleus, and just like a human, it does go through periods of non-REM sleep as well as bursts of REM sleep, and it cycles multiple times over the course of an evening. Well, I guess that these are the cycles of course of an evening. So each of these 20s is sort of a one-day passing that you're seeing across there. And then they have a clear wake period, right? So they have a substantial period when they're awake each day and a period when they're asleep. So they're cycling in a manner that's very reminiscent of what we see in people. And then here's their temperature, and their temperature also cycles just like in people. So they're warmer during the day, and then while they're sleeping, they see about a 1 degree temperature decrease. Here's what happens if you ablate the suprachiasmatic nucleus, though. They're still able to function, like they're still active, more or less, but well, sometimes they're active, but they tend to sleep quite a bit more, and they don't cycle anymore, right? I mean, they're going in and out, but it's not real periodic like we saw over here. There's not a super consistent awake period, except for very occasional, brief periods. Their temperature is just all over the place. They're not cycling like they normally would be. So this is another, piece that's indicative that the suprachiasmatic nucleus is really important in terms of determining our biological clock, our ability to cycle effectively. So how does it work? This is kind of getting into what we did at the homework, but I'll just sort of overview it here, and then we'll revisit it when we get to the homework. And I'm almost out of time. I got 10 minutes, that's right. So here's the idea. If you look at the cells of the suprachiasmatic nucleus, it's not immediately obvious how they can be doing this. They are apparently telling time. They are measuring time in some sort of cyclical way. And the best explanations for how this works is that they employ a suite of clock genes. There's actually at least 3 that have been identified and seem to be particularly important. There's a PER gene, which stands for period. There's cryptochrome, which I don't know anything about. And there's another one that's creatively called clock, which I think was one of the more recently discovered ones. But all 3 of these seem to operate in a similar kind of a fashion, and they're probably all operating redundantly with each other, so you have sort of reinforced information. That's the way a lot of our cells tend to work. The basic idea is that you have a negative feedback mechanism that has a bit of a delay to it. So you'll start at one point in the day, be it at noon or be it at midnight or some other period, you'll start transcribing rather, the clock gene from the gene into its messenger RNA. And then the messenger RNA, as we've seen before, gets translated from messenger RNA into a protein. So that will happen for a while in all these clock genes. You'll be in the process of transcribing and then translating, and you'll be accumulating this protein. But what will happen is eventually, after a certain amount of time has passed, just because of the rate of accumulation of this protein, once you get above a certain threshold, this protein will come back and will inhibit the transcription pathway and stop you from manufacturing more messenger RNA. So if we plot a graph, you'll be cruising along initially, and you'll start to manufacture more and more. I'm just going to choose one. Let's say it's the clock protein. We'll manufacture more and more clock protein until we get above a certain threshold amount, which seems to then start to inhibit the transcription and manufacture more clock protein, at which point you'll start to see a decline, and the thing will sort of dip down, and by the time the inhibition is released, and then you're able to start transcribing more and start translating more and creating more of the clock protein, much of that clock protein has been destroyed or deactivated, and so then you kind of start from scratch again by the time you get that system up and running again. And in this way, you see the protein build up, inhibit itself, crash, build up again, inhibit itself, crash, and the rate at which you're doing this, one period, like going from here to here, this takes about 24 hours to complete. And you can modify somehow, we don't know how this happens, but you can modify the period based on external stimulus, coming out of those ganglion cells up into the suprachiasmatic nucleus. But you seem to sort of turn on these genes, manufacture the protein, build up their levels, and then they inhibit themselves and they crash again. And they seem to cycle in this manner every day. And the result is that we have this little internal clock. I mean, it's a molecular clock. It's imperfect. As we saw before, it maybe is on more of a 25-hour time period than a 24. Other people will vary in different directions. Different animals vary in different ways too. So some animals seem to be biased towards a shorter day. Some are biased towards a longer day like us. But pretty much every organism that I know about has some manner of system that works like this. Even bacteria have a system that works like this, which is pretty cool. At least many bacteria do. In that they'll have some kind of clock system that allows them to have an internal biological clock. Does that make sense? We'll see it again when we do the PER gene in the homework. I think it does a good job explaining it. So if the suprachiasmatic nucleus is doing that kind of cycling, what's going on around the rest of the body? The cool thing is, almost every cell in the body actually has these same kinds of clock genes, PER gene, clock gene, the other one that I'm forgetting the name of. Those genes seem to be ubiquitous. They're not just housed in the suprachiasmatic nucleus, but it seems that the suprachiasmatic nucleus seems to be the main clock keeper. And then it is sending signals to other centers within the hypothalamus that influence all kinds of other body systems, which finally, in turn, inform the rest of our body about what to do. So what this is showing is that the suprachiasmatic nucleus might be setting a basic output. It then is talking to other hypothalamic centers that do other things. So for example, the hypothalamus, we said back in chapter 7, one of the many things it does is that it's basically responsible for regulating our entire autonomic nervous system. It is a thermoregulatory center. So our main thermoregulatory centers are housed within the hypothalamus. It's a major control center for much of our endocrine system. So for example, it releases a hormone that ultimately causes the production of cortisol, which is our stress hormone that we were just talking about a little bit ago. Feeding centers are regulated within the hypothalamus. So our motivation to eat or our motivation to drink, all these sorts of centers are regulated by the hypothalamus. And the suprachiasmatic nucleus is talking to all these hypothalamic centers. And then it influences then their motivations, and so your tendency to be hungry, or your tendency to change your body temperature, or your tendency to change the regulation of internal systems with your autonomic nervous system. These centers are all basically run by the hypothalamus that are getting input from this suprachiasmatic center within the hypothalamus as well. And so then they're varying their signals. And those hypothalamic centers then are feeding out information that go to pretty much all the other organs. So they're showing the liver here, but it's not just the liver. I mean, it's pretty much every other organ within our body is responding to these cycles that are ultimately attributable all the way up into the suprachiasmatic nucleus. And certainly we've all experienced, just one second, I'm going to finish this thought or I'm going to forget. We've experienced situations where the suprachiasmatic nucleus is not getting information that is consistent with our external cues. So if you've ever been jet lagged, which probably many of you have been at some point, if you travel even just a couple hours out of your time zone, we all sort of experience this jet lag phenomenon where, and this is especially severe if you've ever gone like 12 hours worth of time zones out of phase. You'll be basically, your suprachiasmatic nucleus will say, I am awake, and it's the middle of the night, or vice versa. It'll be the middle of the day, and it's telling you that it's basically time to sleep, because its rhythm is not perfectly entrained then with the external environment. And it takes several days to sort of overcome those challenges. So I'm sorry.
This might be a bit of a stretch, but the cognitive awareness that time is passing, the internal clock, do you think this could be influenced by this? Or do you think that's
Yeah, cognitive knowledge of time passing. I have no idea. That's a really good question, and I don't know anything about that. But I like the idea of that. Yeah, I just don't know. I'd be a little skeptical because I don't think we're very good at measuring internally I don't know. Like how we're good. Yeah, I don't know. I tend to think of like us being reasonably good at figuring out if 10 minutes versus 20 minutes, or 10 minutes versus an hour has passed, as opposed to 6 versus 8 hours. But I don't know. But those short time periods, I wouldn't think would be enough to cause much of a change in concentration in the proteins, whereas longer time would. It's a very good question. I just don't know. Other questions people have? Am I done? I'm done. So I'm sorry, I went on too long. There's only 3 minutes left. I think we'll just call it here. I know you did your homework for today, and I apologize. We will do homework on Wednesday. That's going to be our goal for that day. And then we'll probably kick into the last unit when we get back from break.